hello again. This is Dr. Deborah Morris, and we are going to be talking about a whole new set of concepts and ideas in this module. We're going to discuss the physiology of the heart and the vasculature. This is one of the most important parts of the physiology course in terms of your future understanding of clinical medicine because having a good understanding of the physiology of the normal heart and the physiology of heart muscle and of the electrical system of the heart and of the inputs into what happens with the heart, all of those things and clearly an understanding of the electrical system of the heart will help you understand cardiology, help you understand EKGs, which you'll be studying next spring. So this is important stuff, but it's also interesting and fun, and I think you'll enjoy it. Again, because of the amount of material in this PowerPoint and the complexity of the material, I am likely to be dividing this into two recordings. And another thing that I'm going to repeat here is that because repetition is the key to adult learning, there's going to be a lot of repetition as I talk to you about these concepts. And I'll be using different ways to explain the same concepts. I'll be using different diagrams, but you will notice that we'll be coming back to the same things over and over. I want to start by just expressing the idea that as we learn about cardiac physiology, there are sort of three things that you're going to be thinking about and considering. One of those things is pressure. The heart is a pump, and pumps generate pressure, and the pressure generated by the pump that we call the heart is what moves blood through the vascular system. And so a lot of what our understanding of cardiac physiology is going to have to do with pressures. Next, that pressure generates flow. And flow is an important part of cardiac physiology. For instance, the concept of cardiac output, which is how much blood leaves the heart or is pumped through the vasculature in a given time frame. So those are things that we're going to be talking about. But also, there's a lot of important electrophysiology because this pump is operated not by the nervous system, but by its own specialized electrophysiology. And so understanding how that relates to the pressures and to the flow, bringing those three things together will give you a very good understanding of cardiac physiology. You all know that the heart is a pump, but really it's two pumps, and it's two pumps in sequence or in series. The right side of the heart accepts deoxygenated blood from the vena cava under very low pressure conditions. So blood flowing from the vena cava into the right atrium is at only a couple centimeters water of pressure. It is a low pressure system. It then remains under relatively low pressure as the ventricle contracts, obviously raising the pressure because that's how pumps work, and ejects that blood into the pulmonary arteries. Remember that on the right side of the heart, we're dealing with deoxygenated, poorly oxygenated blood that is returning to the system, and that we are pumping it into a relatively low pressure vascular bed in the pulmonary system. So the blood is pumped through the pulmonary artery and its branches into capillaries that are in contact with the alveoli of the lungs, where this deoxygenated blood is allowed to pick up oxygen. Tends to be relatively low pressure, tends to be relatively slow flow, so that there is time for the blood to release carbon dioxide and pick up oxygen. And then that blood is returned to the left side of the heart, which is its own pump. It's still under relatively low pressure, but higher pressure in the left atrium than it was in the right atrium. And the left ventricle generates pretty high pressures in order to push that blood to create blood flow through the systemic vasculature, specifically into the aorta, up to the brain, to the kind of tip of all of our limbs, through all of our organs, including the kidneys, which are really crucial. So, this is a higher pressure system. And on the left side of the heart, when everything's working normally, the blood is oxygenated. So, low oxygenation on the right side, low pressures on the right side. Oxygenated blood, high oxygen on the left side, high pressures on the left side. The flow of blood from this series of pumps goes through arteries and veins. And there are some, so we'll come back to this a little bit later. So I'm not going to go into tremendous detail, but the blood initially is pushed into elastic arteries with thick walls with a number of layers, including quite a lot of muscle and an endothelium or lining and an outer layer of connective tissue to provide a lot of structure. Um, and then through increasingly narrow arteries. So the number of arteries is going to be increasing, but the diameter of those arteries is going to be decreasing. And as we go from these big elastic arteries down to the small arterioles just prior to the capillary bed, you notice that we lose a lot of the layers, but we always continue to have an endothelium, which is the cells that line all of our vessels, and smooth muscle. The capillary bed consists of vessels that are a single cell thick, meaning that the only layer that's left when you get to the capillaries are the endothelial cells. They are surrounded by a structure called basement membrane, which is not cellular. There are capillaries that are continuous, and there are capillaries that are called fenestrated or sort of leaky, where there are small pores in the endothelial cells that allow freer movement of fluid in and out of the fenestrated capillary. Continuous capillaries, there is some diffusion of gases across the endothelial cells, but there's not a lot of movement of fluid. In fenestrated capillaries, there is. And then as we get to the venous side, you see again that they increase in diameter, they increase in structure. But there is always a lining called endothelium. And in medium sized veins and large veins, there is smooth muscle. All of that smooth muscle has the ability to contract or dilate depending upon signals that it's receiving from the autonomic nervous system and also from circulating hormones like epinephrine. So first time through, we're going to enter the right side of the heart. The blood comes into the right atrium from the vena cava. There is no valve between the vena cava and the right atrium, so the blood flow is pretty continuous, and that blood is deoxygenated. It has lost a lot of its oxygen. It's carrying a certain amount of carbon dioxide, which is a waste product of metabolism. That blood moves through a valve called the tricuspid valve from the right atrium to the right ventricle. When the right ventricle is full and the electrical activity causes it to start to contract, the tricuspid valve closes so that there is not, under normal circumstances, backflow. And all of that blood is pushed forward out of the right ventricle into the pulmonary artery through a valve called the pulmonic valve. That blood then is distributed throughout the pulmonary capillary bed, surrounding the alveoli or the air sacs of the pulmonary system. And here it collects oxygen and it loses carbon dioxide. And the oxygen is diffusing in and the carbon dioxide is diffusing out. How they are carried is something we'll talk about later. But the blood then collects into pulmonary veins. And unlike the veins in the systemic circulation, this is now oxygenated blood. And it is returned through the main pulmonary vein into the left atrium. And again, there is no valve. And that oxygenated blood now is in the left atrium and will move into the left system. So the oxygenated blood in the left atrium flows through a valve into the left ventricle, and that valve is called the mitral valve. Once the left ventricle is full and electrical activity causes it to start to contract, the mitral valve closes and the blood is ejected through a valve called the aortic valve into the aorta. It then flows throughout the body into systemic capillary beds. So those capillaries are everywhere. They're in the brain, they're in the nose, they're in your fingers, they're in your kidneys, they're in your fatty tissue in your skin. And throughout that flow, it loses oxygen in the capillary bed and it picks up CO2. And again, the oxygen and the CO2 are both gases that are lipid soluble and pretty much freely diffuse across the capillary walls, the endothelium, the cell membrane of the endothelium. And then that blood is returned to veins and then the vena cava and comes back to the right atrium, deoxygenated. So that's our circuit. We'll come back to that circuit and we'll do it a few times, but I want to talk a little bit about definitions. These are terms that you have to understand in order to understand cardiac physiology and cardiology. So the first of these terms is a concept called cardiac output. It is abbreviated as CO, and it's the amount of blood that is ejected by the ventricles into the pulmonary circulation and the systemic circulation. It's about the same amount per unit of time, and it's usually going to be stated in liters per minute. Blood that returns to the atrium, the right atrium, or the left atrium from the systemic and pulmonary circulation is called venous return. There is a concept that's a little bit harder to understand called preload. And preload is essentially a concept describing the volume and pressure in the ventricle at the end of diastole, so just before the ventricle contracts. Um, and so it's dependent on how much blood is coming back, so venous return, and the pressures of that blood in the left ventricle. We don't usually talk about right ventricular preload. We are thinking about preload as a concept regarding the left ventricle. So it has to do with the amount of blood that has returned into the cardiac circulation through venous return. So that's what's coming into the right atrium and going through the lungs and then coming into the left atrium, along with the pressures at the end of diastole, just before um, the ventricle contracts. So we could call it end diastolic volume, but pressure is important here too. So that's preload. Afterload has to do with the pressure against which the ventricle is ejecting the blood. So when the aortic valve opens, you have to think about the fact that there is a pressure in the systemic circulation. At the end of diastole, it's diastolic blood pressure. And at the end of systole, it's systolic pressure. So afterload is the pressure against which the ventricle is ejecting blood. There is something called stroke volume, and this again usually refers to the left ventricle, and it's the amount of blood ejected with each ventricular contraction. And there's a concept called ejection fraction, which is the percentage of the blood in the ventricle that's ejected during each systolic contraction of the ventricle. So when the ventricle contracts, it does not eject 100% of the blood in the ventricle. It may eject 70%, which is good. It may eject 20%, which is crappy. But that's the ejection fraction. And here we have a few more definitions, and we'll come back to some of these as we go through this presentation because they'll be helpful. So first, there are two sets of valves, right? There are the valves between the atria and the ventricles. On the right side, it's the tricuspid valve. On the left side, it's the mitral valve, and they're called AV valves. You will occasionally hear the mitral valve, which is between the left atrium and the left ventricle. You'll hear it called the bicuspid valve. Then there are the valves out of the ventricle into the pulmonary artery and the aorta, into those big arteries. And they are called the semilunar valves. So the aortic and pulmonic valves exiting the ventricles are the semilunar valves. Systole is the time during which the ventricle is contracting and generating pressure. Diastole is the period of time during which the ventricle is relaxing and filling with blood. You've all listened to somebody's heart with a stethoscope. We do it, in childhood, we play stethoscopes. We do it at the doctor's office if we have a friendly doctor. And then, of course, we do it as we start to enter the health professions. And when you listen to the heart, you generally hear lub dub, lub dub, lub dub. Well, we define those sounds. We give them names. And S1 is lub. It's the first heart sound, and it is primarily generated by the closure of the mitral valve. The mitral valve closes and you get a vibration, lub. The second, and that's at the pretty much the beginning of systole, the end of diastole, because as soon as pressure starts to rise in the ventricle, it pushes the mitral valve closed. The second heart sound, or S2, is generated primarily by the closure of the aortic valve, and it's dub. It's a little more complicated than that because the closure of the pulmonic valve is also pretty loud, and depending on what part of the chest wall you're listening to, S2 may have two components. So S1 is closure of the mitral valve. S2, second heart sound, is closure of the aortic and pulmonic valves, and they are lub and dub, lub dub, lub dub. Now there are two additional sounds that we call heart sounds that are abbreviated as S. There's something called S3, which is the third heart sound. It is sometimes heard as the ventricle fills. In kids, it can be normal, and in young people, but in adults, it usually means there's something wrong, and that something wrong is usually that the ventricle is enlarged. And the chordae tendineae, which are these sort of strings of heart muscle that stabilize the mitral valve, tense, sort of like a guitar string. So you get a vibration from the tensing of the chordae tendineae. And an S3 in an adult may indicate the possibility of congestive heart failure. S4, or the fourth heart sound, is a heart sound that is sometimes heard during the period before, like at the end of diastole, just before ventricular contraction. Actually, S3 is usually heard during filling, so early diastole. S4 is late diastole. As the atrium contracts right before the ventricle contracts. And it happens when the ventricular wall is stiff, is not compliant. And that can happen if it's thickened, like ventricular hypertrophy, or occasionally if it's ischemic, because ischemic heart muscle is less compliant or less stretchy than heart muscle that's getting lots of oxygen. And it's another indication of abnormal heart function and possibly of ongoing heart failure. There are two areas in the heart muscle of specialized cardiac myocytes that we call nodes. One of them is the SA node, sinoatrial node. It's located in the right atrium, very close to the vena cava, and it's the primary pacemaker of the heart. So the SA node is the pacemaker that generates the electrical activity that results in the beating of the heart. The AV node is located low in the right atrium, and its function is to slow conduction from the atrium to the ventricle, but it can also function as a secondary pacemaker. So sometimes when the SA node quits working, you may end up with a slower heart rate that is generated by the AV node. Heart muscle is interesting. It is a little bit different than smooth muscle, but it has some similarities. It's a little bit different than skeletal muscle, but it has some similarities. So they are myocytes. They are striated like skeletal muscle. And the striations are the filaments of proteins called actin and myosin that are sliding to contract and to relax. And we'll talk more about how that happens later. But actin and myosin filaments slide to contract and to relax. One of the things about the heart muscle is that it functions as a syncytium. So each cell actually has direct communication with the next cell. So these cell membranes are fused by something called intercalated discs. And then you have kind of series of myocytes and parallel myocytes, and the action potentials that are generated initially in the pacemaker in the SA node are allowed to propagate from cell to cell directly so that the cells contract together as a unit. There is a non conductive kind of fibrous layer of tissue between the atria and the ventricles to keep them from all contracting together. For maximal um, function of the pump, the atrium has to contract first. And if it was one syncytium, you'd have the whole thing just sort of starting to contract and contracting together. So there is this fibrous non-conductive tissue between the atrium, both atria, and both ventricles that prevents direct communication of that electrical impulse from the atria to the ventricles, except at one point, which is the AV node. And then from the AV node, there are tracts of specialized myocytes, muscle cells, that function similarly to nerves and conduct action potentials. Those tissues, which we call the bundle branches and Purkinje fibers, can also, under the um, abnormal circumstances, they can actually also generate action potentials in the same way that the SA node and the AV node can. So they can end up functioning as pacemakers if the SA node isn't functioning and the AV node isn't functioning. Now, while the heart is clearly innervated, and it's innervated by fibers from the autonomic nervous system, both sympathetic and parasympathetic, something that's really important to understand is that if you cut the nerves to the heart such that the heart loses its innervation, it just keeps right on working. Now, it's not getting signals from those two systems, the sympathetic and parasympathetic arms of the autonomic nervous system, to help it beat faster or slower to maintain proper homeostasis, but it's going to continue to generate action potentials and to contract so that the heart will keep beating. You can cut the nerves to the heart and it will continue to beat. Here are some diagrams just illustrating cardiac muscle. This one's a bit of a combination of a drawing and a micrograph. It's showing these cardiac cells, the intercalated discs that allow really direct flow of electricity and I believe also cytoplasm from cell body to cell body. There are lots of nuclei. There's a little bit of branching. In here, this is a diagram of the same thing, showing the striations of actin and myosin, showing multiple nuclei because there are multiple cells, but they're connected at these places called intercalated discs, and they function as a syncytium. They function all together. This is a diagram showing the inside of a myocyte. Here we have the connection of the cells in their syncytium at something called a desmosome. You can see the fibers of actin and myosin that are interacting to allow the contraction and relaxation of the muscle. There are within myocytes, and this is smooth muscle, skeletal muscle, and cardiac muscle. What we would call endoplasmic reticulum in other cells is called sarcoplasmic reticulum. And a big part of the function of sarcoplasmic reticulum is to collect calcium ions, and then there are voltage gated calcium ion channels that allow the release of calcium ions when there's an action potential. And it's calcium interacting with these actin and myosin fibers, the cardiac myofibrils, that create the contraction of the muscle. There are also structures called T tubules where the cell membrane, the sarcolemma, actually extends deep into the cell, again allowing an action potential flowing along the cell membrane to get within the cell and interact with the sarcoplasmic reticulum. This is something we'll come back to much later in the course when we talk about the muscle um, neuromuscular function and the skeletal system. But it's showing you these proteins interacting with each other. There is something called a thick filament, which is made up of myosin, something called C proteins, and has these little arms on it. And there are thin filaments that are made up of actin and other proteins, including tropomyosin and troponin. When calcium is released from the sarcoplasmic reticulum, it activates these arms of myosin, which grab on from the thick filament to the thin filament and pull the whole thing together, contract. And when we're looking at the striations um, in skeletal muscle and cardiac muscle, what we're seeing there are the places where we have either overlap of the thin and thick filaments or lack of overlap of the thick and thin filaments, along with the proteins to which they attach that are called Z lines. And that's all we're going to say about muscle at the moment. I do want to talk a little bit more about the electrophysiology of the heart and how it is generated and how it is conducted. So I said earlier that the SA node, or the sinoatrial node, which is the primary pacemaker of the heart, sits in the right atrium quite close to where the superior vena cava enters. That node is specialized myocytes, so it's cardiac muscle, but it's specialized cardiac muscle that has something called automaticity, and that automatically, periodically depolarizes. And when it depolarizes, it creates an action potential, which then passes through the syncytium of cardiac muscle that forms the right and left atria, causing contraction of the atria, of the right atrium and the left atrium. When that electrical impulse gets to this fibrous base, it does not continue to flow into the ventricular muscle. What it does instead is it gets picked up at the AV node, at the atrioventricular node, and it's slowed down a little bit as it flows into the bundle of His, again, specialized myocytes, but functioning a little bit like nerves and carrying that action potential. Then to the bundles, the bundle branches left and right, and then into fibers that extend throughout the ventricular muscle that are called Purkinje fibers. So this impulse coming through this tissue now causes contraction pretty much all at the same time of the ventricle. So in the atria, it's just spread through the syncytium, the impulse. In the ventricles, it comes through the AV node, the His bundle, the bundle branches, then out to all these fibers called Purkinje fibers that are spreading throughout the ventricular muscle and causing near simultaneous contraction of the ventricular muscle, so depolarization. Again, without innervation and the innervation is to the sinoatrial node, but without innervation, the heart's going to continue to beat and contract. Now, without innervation, if you get attacked by that tiger, you're not going to get sympathetic impulses directly to the SA node to speed it up. So you're not going to have that help in maintaining homeostasis and in fight or flight. Ultimately, you will get some epinephrine through the circulatory system functioning as a hormone, so your heart will still speed up a bit, but it's not as fast, it's not as effective. And we have reached what I consider to be sort of the center of cardiac physiology. This is a diagram that was initially developed and has been modified, and there are just a million different variations. But the guy was a cardiologist named Dr. Wiggers, and the diagram is called the Wiggers diagram. And the Wiggers diagram illustrates a few things. The primary part of the Wiggers diagram is up here, and on the x-axis is time, and on the y-axis is pressure. And as time goes through a cardiac cycle, we are illustrating changes in pressure in different places. We are looking at the left atrium, we are looking here at the left ventricle, and we are looking at the aorta. Now, we could add other things. We could add other pressure curves here. We could add right ventricular, right atrial pressures, and there are times where you will see Wiggers diagrams that have all of those pressures. Also, on the Wiggers diagram, we have added some other things. One of the things that is added, and again, this axis is time. It's the same time. But we have other events that occur in the heart other than pressure changes. Remember, I said we have electrical changes. So the electrocardiogram, the electrical changes that occurred during the cardiac cycle, can be plotted against time on the Wiggers diagram. And understanding the relationships between these events and these events is important. We can plot a phonogram, heart sounds, and here we have S1 and S2 plotted. We also show S3 and S4, which could occur. And we are here plotting something called the venous pulse, which is roughly the pressures in the right atrium, and they change during the cardiac cycle. The venous pulse we can observe under certain circumstances in the neck, in the jugular vein, particularly on the right, and we'll come back to that in a little while. Now, there are a whole bunch of other things on here, right? There are descriptions of events. So as the cardiac cycle is going along here, we have the P wave. The P wave causes contraction of the atria. And this venous pulse, the A part of the venous pulse, is from the contraction of the right atrium. Okay? But a time comes where we have the QRS complex, which is, this was atrial depolarization. This is ventricular depolarization. And when the ventricle depolarizes and starts to contract, suddenly the pressure in the ventricle is higher than the pressure in the atrium, and the mitral valve gets pushed closed. And now we have ventricular contraction. And when the pressure in the and again, we are really here talking about the left side. Down here I'm talking about the right side, but it all works together. When the pressure in the ventricle exceeds the pressure in the aorta, then the aortic valve opens. Sorry. These periods of time, different things are happening. Okay. So during the period where the P wave causes atrial um, depolarization and atrial contraction, we have something called atrial systole. When the mitral valve closes, that's the end of atrial systole and the beginning of ventricular systole. So ventricular systole goes from here, this line, to this line. The ventricle is contracting. Blood begins to flow into the aorta when the aortic valve opens, and you can see there's this little period of time where um, we hear the mitral valve close, so there's a vibration, S1, and the ventricle is contracting, but the volume isn't changing. So pressure is going up very quickly, and that is sometimes called isovolumetric contraction. Once the aortic valve opens, you get rapid ejection. A lot of blood is flowing out at pretty high pressures. Eventually, much of the blood has been ejected, and flow is a little slower. And when the pressure in the left ventricle becomes lower than the systolic pressure, the pressure in the aorta, systolic pressure or afterload, then the aortic valve closes. And we have a period of time where the muscle is relaxing, volume is not changing, pressure is dropping rapidly. And that's called isovolumetric relaxation. Now, during systole, ventricular systole, there is continuously some filling of the atrium. So the atrial pressure is going up a little bit. Okay. Remember that aortic valve closure results in the dub, S2. So suddenly, when atrial pressure exceeds ventricular pressure, the mitral valve is going to open, and you're going to get a lot of blood entering the ventricle. And so there's a period of rapid ventricular filling. If you're young, or if the ventricle is dilated from congestive heart failure, you might get an S3 from those chordae tendineae kind of twanging from the rapid ventricular filling, stretching them. Okay. As time goes on during diastole, you get a slower ventricular filling until the P wave happens, right? And then that P wave causes atrial contraction, which pushes just a little more blood into the ventricle, resulting in the possibility of an S4 if the ventricle is stiff and non-compliant. So that is what we're seeing on this Wiggers diagram. Again, we could add lots more lines, but these are the ones that I think if you understand the relationship between the electrical activity, atrial depolarization and repolarization, ventricular depolarization and repolarization, okay, and the muscle contraction that occur in the atria and the ventricle, resulting in the pressure changes that cause valves to open and close, and the sounds that result from valve closure. If you can put all that together, you're going to understand clinical cardiology. If you'll understand the normal, you'll have a much better understanding of the abnormal. This is the Wikipedia Wiggers diagram. Um, it is showing an additional line. It's showing some things that you're not seeing on the previous one, and that is ventricular volume. So again, this is left ventricle aorta, um, left atrium pressures, left ventricular pressures, aortic pressures, the same pressure on the vertical axis, time on the horizontal axis, but we have an additional vertical axis here for ventricular volume that's showing you that as the ventricle contracts during this contraction, when the valves on both sides are closed, volume stays the same. During the first part of ejection, um, a lot of blood leaves, so volume goes down rapidly. During the second part of ejection, it's a slower flow. Again, when the aortic valve closes, resulting in the second heart sound, you're going to have this period of um, isovolumic or isovolumetric relaxation with the same volume. Isovolumic. And then the AV valve opens and you start to get atrial filling. All right. So all those things that happen from the beginning of one heartbeat to the beginning of the next heartbeat are called the cardiac cycle. And they start with this spontaneous generation of an action potential in the SA node or sinus node or sinoatrial node, which is why we look at it's why we talk about a sinus rhythm, because a normal person's heart that is beating as a result of the pacemaker in the sinoatrial node has a relatively regular heart rate, usually between about 60 and 100 beats per minute, and that is called a sinus rhythm. Okay, so that action potential causes depolarization to spread through the atria, and then as those myocytes depolarize, they contract. That pushes a little bit of extra blood through the open tricuspid and mitral valves into the ventricles. Before that, those valves were open, but there was just passive filling. Blood was flowing in from the vena cava through the tricuspid valve into the right ventricle without anything pushing it. But once the atrium depolarizes and the atria contracts, it's pushing a little harder. Okay. Then the depolarization kind of ends in the atria and they start to repolarize, but the action potential is picked up in the AV node and then spreads, depolarizes through the His bundles and the Purkinje fibers, and that introduces a delay, and the delay is about 1/10 of a second. So the ventricles contract a little bit after the atria quit contracting. So the atria contract first, there's this 1/10 of a second delay, then the ventricles contract, and that closes the AV valves. The AV valves being the valves between atria and ventricles, the tricuspid and the mitral, and opening the semilunar valves, which are the valves between the ventricles and the big vessels, the pulmonary artery and the aorta, the pulmonic valves and the aortic valves. Okay. During relaxation, that's diastole, and most of the time we're talking about ventricular diastole. And systole is the period of contraction. So, one of the things that I think will help you in studying this material is to think through the cardiac cycle as you are looking at the lines on a Wigger's diagram. Kind of go down each line, go across through the time cycle, um, pay attention to the relationships between events. So, for instance, pay attention to the relationship between the electrical activity and the pressures that illustrate contraction of the atria and the ventricles. Pay attention to valve opening and closure, which has to do with the relative pressures between the atria and the ventricles, or between the ventricles and the semilunar valves. Pay attention to the relationships between the relative pressures on the right side, which are lower, and the left side, which are higher, and the systemic circulation, which we call blood pressure. And then pay attention to the heart sounds, to when they occur in the cycle, and what are the relationships, what triggers those heart sounds, which you know is valve closure. And valve closure is triggered by pressure changes. So try to put that all together. Um, this is a kind of busy slide that I'm not going to go through in detail because it's just a Wigger's diagram, right? But it's showing a few more things. This one is showing our electrocardiogram and our heart sounds, our phonocardiogram way up here. It is showing aortic, left ventricular, and left atrial pressures, which we've seen. It's showing ventricular volume, but it's also showing blood flow in the aorta. It's showing pressure in the jugular vein, which we saw earlier, jugular venous pressures. It's showing pressures in the carotid, and you see these little bumps. This is called the dicrotic uh, notch, which is a little bump in pressure when the aortic valve closes. And then radial pressure. So the pulse, this is basically carotid pulse and radial pulse. And you can see radial pulses are later than and lower than carotid pressures and pulses. And then pulmonary arterial pressures and right ventricular pressures, which are on a pressure scale, and just helpful to notice how much lower these pressures are. So at the peak of right-sided systole, right ventricular and pulmonary pressures are about 20 millimeters of mercury, whereas at the peak of systolic of left ventricular systole, ventricular and aortic pressures are at about 120 millimeters of mercury. So big differences. When I'm saying that the right side is a low pressure system, I mean it. And all of this is taking place in about a second, or a little less than a second. And again, this is the Wikipedia Wigger's diagram for you to spend a little bit of time going through, looking at, and establishing your understanding of the relationships. I'm going to stop here, and then we're going to exercise repetition and talk about these things using different pictures, slightly different way of putting them all together.